This project has components of both biology and computer science, so I'll explain some of the biology first with chromosome conformation capture, what Hi-C data is. I'll go into my neural network models, HiCARN, the architectures, our methods that we use, some results and conclusions as well. So chromosome conformation capture is a method that's used to predict three-dimensional chromosome structures inside of the nucleus. So when the cell is not in replication, so most of the time, chromosomes are in this like spaghetti-like structure. They're not in the X structure that you would normally see them as. And we know that structure is directly correlated to function in biology and chemistry. And so if we zoom in on a chromosome here, we can identify topologically associating domains, which are regions of chromosome clusters or chromatin loop clusters, I should say. And you zoom in even further and we get insulated neighborhoods, which are regions of chromatin loops where biologically significant interactions are happening, where DNA transcription is occurring. And so Hi-C data is collected via the Hi-C technique, where the DNA is allowed to cross-link with itself. It's then cut with a restriction enzyme. The ends are then marked with biotin to be ligated together, and those are then sheared to create these things called pair reads. And the amount of interactions between those pair reads are known as interaction frequencies, which construct a square matrix of these chromosomal regions, of these pair read regions, where the rows and columns are equal lengths of the DNA regions of the DNA reference sequence. So what this looks like visually, this first row here is a heat map of the matrix where entries of higher magnitude are shown in darker red, darker colors, where areas of lower magnitude are in white or the lighter colors. So zooming in on these entries, we can identify the regions of significant interaction. So if we zoom in on an area, we can find subcompartments. Zoom in even closer, we get the TADs and other domains as well. And then zoom in on two high contrasting entries, we can identify those chromatin loops where DNA transcription is occurring. And these data come in different resolutions. So we have high resolution, which is anywhere less than or equal to 10 kilobase pairs in length. So that means each column and row represents 10,000 base pairs. Low resolution is typically from 25 to, or anything greater than 25 to 40 kilobase pairs. However, as you can see here we have low resolution data. To the right is high resolution data. You can see low resolution has a significant amount of entries that are lost, whereas in the high resolution we can identify many more interactions that are occurring. So there's a problem here. High resolution data are much more difficult to obtain due to computational and financial costs. So to solve this issue, deep learning models are used to predict high resolution data from low resolution inputs, essentially cutting the cost for obtaining high resolution data. However, these models are limited in their training time and predictive accuracy. So the goal of this study was to develop a novel enhancement algorithm to improve upon the limitations of those existing models. And so my model, I don't have time to go into all of the competing models, but mine is a cascading residual network, which was proposed by An and Sohn in 2018, so it's relatively new. And it uses a sequence of 3 residual blocks that incorporate local and global cascading connections. And most of that probably meant absolutely nothing to many of you, so I shall explain. A residual block is simply a sequence of convolutional layers and ReLU activation functions, where a convolutional layer is simply a filter that runs across the matrix and extracts prominent features from the matrix. So that would be entries of high or low or entries of higher load magnitude. And then the activation functions simply just map the outputs of a convolutional layer to a given range. So for example, the ReLU maps anything greater or less than or equal to 0 to 0, anything greater than 0 from 0 to infinity. And the residual blocks incorporate skip connections, which essentially help to force the model to learn a bit more. So there's a problem with deep learning models called the vanishing gradient problem, where essentially they think that they have the right answer when in reality they don't. They think that they've learned enough, but they don't. And I'm using think very strangely here because they don't think, but what it does is it reintroduces that low resolution input over and over again so that the model thinks that it doesn't know enough. And so it will continue to broaden its learning or continue to learn. And so I used two different models of HiCARN. HiCARN 1 is a sequence of 5 cascading blocks that extract information, prominent regions from the high resolution data, and it learns how to convert low resolution data into high resolution data. Whereas HiCARN 2 has the same, what's called a generator, same generator architecture as HiCARN 1. However, it incorporates a discriminator which acts to classify the predicted high resolution data from the real high resolution data. And all this does is it again, like the skip connections, forces the model to continue to learn a bit more. So if the discriminator misclassifies the two as being the same image, then the generator is punished, the model is punished. So it's in what's called loss. So it forces the model to learn a little bit more so that it doesn't think that it's fully right. And then for our methods, all of our data was taken from data collected from Rao et al. In 2014. And for our low resolution inputs, we use 1 out of 16 to 1 out of 100 randomly downsampled high resolution matrices. For training, we use the human GM12878 cell line, which are lymphoblastoid cells, using a holdout cross-validation. So all this means is that 4 chromosomes were left out during training to test the model's accuracy across the training session. And then for testing, we tested the remaining 4 human GM12878 cell line chromosomes to test across cell lines. We did the human K562, which are leukemia cells, and then also we tested across species to the CH12LX mouse embryonic stem cell line. And then for our analysis, we used image similarity metrics, which they're pixel-wise comparisons between the high-resolution image and our predicted images, and as well as high-C reproducibility, which are stratum-adjusted correlation coefficients that were developed specifically for these resolution enhancement models. And then biological validation as well to ensure that our predictions are consistent with the ground truth data, so are able to detect those biologically significant regions accurately. And so these results, I don't have time to show them here, and they're all box plots anyways that you don't want to see. For image similarity, overall, our model outperformed the existing models in all of our metrics. For high-C reproducibility, we used 2 different tests, genome disco and high-C rep, where our model outperformed in genome disco. However, high-C rep, there were no differences across the models. And then for our biological validation, we outperformed in T80 predictions, and our model, as well as DeepIC, maintained high performance throughout the chromatin loop predictions. And our model also maintains high performance and with low training times. And to give you a visual of what our images look like, here we have some competing models over here, but what I want you to focus on is this high resolution image. That's the target that we're going for. We have HiCARN 1 and HiCARN 2 that produce very visually similar images to the high resolution data. So this 1 out of 16 downsample is run through our model, and this is what is reproduced. And in the 3D chromosome structure predictions, this is what it would look like. This is how the heat map converts to a 3D structure prediction. So you can see to the left, that's the low resolution image in white. You can't detect any significant regions from that prediction. There's nothing significant going on there. However, our models, even though our predictions are not perfect, they're still much, much better than the low resolution because you can see these areas of significant interactions going on, specifically right here where there's a topologically associated domain where researchers can identify where DNA transcription is likely to be occurring. And then this is across cell lines. As a similar thing here, HiCARN 1 and 2 reproduce very visually similar images to the high resolution data. Other models produce somewhat blurry images. However, high CSR and DeepIC do pretty well as well. And then this is across species, and this is where you really start to see the robustness of our model, where HiCARN 1 and 2 are able to accurately reproduce the high resolution image, whereas all these other models do not, for the most part, maybe HiCSR, do not do it as well as we do. And then for our topologically associating domain boundary predictions, as you can see here, the low resolution image is not very sufficient at identifying those those significant interactions going on. So the black line you see here is the boundary of a topologically associating domain. And the goal is to reproduce this high resolution image here, those boundary lines. HiCARN 1 and 2 do that very well. DeepHiC also performs pretty well. However, HiCSR, HiCNN, and HiC+, those are some other existing models, they do not necessarily do it as well as we do. So in conclusion, overall, our model outperformed all other enhancement models across metrics and biological validation. We found that the variations in our framework, HiCARN 1 versus HiCARN 2, did not significantly hinder or improve the performance of the model. It maintained consistency throughout. And then our model just further contributes to the enhancement of low resolution data to high resolution data to effectively cut the cost of obtaining high resolution Hi-C data. I would like to thank my advisors, Dr. Sarah Karam and Dr. Oluwatosin Oluwadare, as well as Sean Bignami for his comments. Do we have any questions?